This
usual thing
conference
of
will
pressing
now be
record.
recorded.
Excellent. Okay, well, hello everyone, and welcome to this second seminar of the Ideal for OMOP training course. So hopefully you can all see me, you can hear me, and also you can see the screen that's being presented. So Will, if you'd be kind enough to go into a presentation mode and just take us to the introduction slide, slide 2. Oops, and we've lost it. Right, well, while Will's rediscovering that, hopefully in terms of introductions, there'll be starting to be some familiar faces on the calls now. So I'm James Anderson. I run and oversee the DIGICORE training programs, of which this is one. And Will as well, I think you may recognise, Will has been closely involved with the running of this program and some of the antecedent programs as well. It's also a great pleasure today to welcome two perhaps slightly newer faces who will be doing the majority of the presenting today. So we've got Atif Adam, who is from the IDEAL4OMOP team, an expert epidemiologist and veteran of many studies in OMOP, and also Rosie McDonald, who is been able to lead and support a number of the current OMOP studies that we're doing through DigiOne. How are we getting on with sharing those? Your screen, Will. In fact, while you're doing that, the other request to you was, again, as we did last time, if you could just say hi, say who you are, where you're from, which institution, if you have a particular tumour specialty, and also just because we're British and we're nosy, anything about where you are, maybe what you're going to have, looking forward to having for dinner tonight. It's always nice to see a little bit of different cuisine from around the world. Right, we're still struggling to present.
I'm in a meeting room here with Will, who is sharing his screen. We now load it. It's not clear why. He's seeing it on his screen and it's taking a bit of time to load. So, but we do now see, if you go to full screen.
Brilliant. And if you could perhaps go to slide 3, that would be awesome. When we get to that, I'm just going to remind you very briefly where we currently are in the program. So we recruited, and it's brilliant, as we shared last time, we've got over 70 people who've signed up to the course, which is absolutely fantastic news. And we're now in that first phase, which is we're calling basic training and team formation. And in that phase, there's going to be a series of training events like this, starting to equip you with the tools, the knowledge, some of the key concepts around studies in OMOP. But also, as we move towards the summer, starting to encourage you to work together in teams and starting to develop your own prototype scientific studies. As we go into the latter part of the year, you will have an opportunity to apply for funding for those multi-site studies and actually get going and produce some real analytical outputs as we get into 2025, building off the data from your own organizations and the organizations and the teams that you put together. So that's sort of where we are in the program overall. But right at the moment, we are in that talk component, and this is actually the second of 6 seminars that we're running. So I think many of you were in the seminar 2 weeks ago, which was an introductory seminar to some of the key concepts of OMOP. Today, we're going to start to talk about studies in OMOP and database studies and share some insights around that. Next week, we've got a seminar looking at some of the more technical aspects of OMOP and what the implications of that are for our studies. And then later in the month, we're looking at patient involvement. And as we get towards the summer, we'll be sharing seminars on data privacy, and then also finally preparing for that team, the team formation. So that's all well and good. It may be that some of you are kind of super confident already, and you've already got a clear idea of perhaps how you might start to put together a team, what the study you might want to do looks like, and that's great. I suspect there's probably also some of you out there who are thinking, golly, that sounds like quite a big ask from where I am now to actually, conducting a study using using OMOP standards, building off the data that we have in our organization. So if I can just try and put your mind at rest around that, there is going to be a lot of support for you. Now, clearly, these seminars are part of that support, but they are only part. So there's 6 seminars. They've been designed and created specially for this course to be as relevant as possible and make, pitched at the right level. That's not the only thing. So later in this call, we'll be demonstrating, I hope, a course website. It's a SharePoint site, which will be a repository of lots of different resources, these webinars, other kind of online resources, all sorts of things that you'll be able to go to and have as something of a library. On top of that, you should, from now on, really feel free to reach out to the training team. That's me and Will. If you've got specific questions or you want to have some more content, some more support on particular topics, we can help provide that for you. But as I say, whether it's specific questions or I'd just like to know more about X, Y, or Z, you can have that. As we get to the latter part of the year, you'll remember there's a leadership advisory board of kind of really internationally renowned experts in this field, and we will have time with them. You will have time to go to them with questions, with thoughts, and with proposals for your studies. So that will be another important component of the support that we supply to you. Actually, the last bit of support in some ways will be the most important, and that is each other. So we've got a great range of experience of people from different organizations, different backgrounds. Some of you have already got experience with OMOP as well, many of you with real world studies. So as you start to get to know each other, you'll be able to learn from each other and rely on each other and build on each other's knowledge and expertise. So that's all really by way of introduction. Are we still suffering from a bit of lag on the slides? Because I can only see slide. Yeah, okay. Can we go on to the next slide, perhaps? There we go. Thank you. So objectives for today, as I said, the majority of today will be hearing from Atif and Rosie, and we'll be looking at the different types of studies that can be carried out, just to give you a flavour of some of this, some quite simple through to some really quite complicated studies. We'll reflect a little bit on some of the challenges that these studies can present, and then we'll switch over to Rosie, who's going to be talking about a study that we've recently run within DIGICORE, within what's called the DigiOne group, and some of the specific learnings we've had there. So hopefully just to give you a chance to see how it might work in practice. And then we'll wrap up at the end, and all being well with the technology, we'll tell you a little bit more about some of the resources at your disposal, but particularly the SharePoint, that website, that course website that will have lots of this information. So that was all I wanted to say. I suggest now we pass over to Atif, who's going to talk to us about database and OMOP studies.
Thanks so much, James. Can everyone hear me? Fine?
Yeah, I can hear you.
Yes, we
Perfect.
can.
Awesome. So as I kick this off, I think if there was anything you were to take away, if you were to zone out, it's this first slide that I want you to keep in mind. And there's another slide coming up, which I won't tell you what that slide is. You just have to pay attention to me. But the idea is, as you build studies, it's important for you to consider a balance between what you have and what kind of study you want to do. Realize that it's not reaching the final line that you're interested in. It's kind of building processes and complexities in phases, and how you can leverage the OMOP CDM and the HEDOC MVP to really get you there. And the fact that as a team, just like James mentioned, we're working on this together. There's folks with the same competency, they're experts, they're people who are just coming in. How do we leverage each other's strengths to build this consortium and group together? If we're on the same page in that, I think as we go through these slides, everything becomes really simple. I do want to highlight a lot of these slides from the Odyssey communities. I do want to give a shout out to everyone in the oncology working group and the other work groups who contributed to some of the CDM work and everything happening to make OMOP a viable CDM that's globally recognized as it is. Next slide, James. So when we think of real world evidence, we really think of reproducibility. Right? The idea is that if you want to do a study, can other folks repeat the same amount of work? At what scale are we talking about reproducibility? Depending who your stakeholder is, it regulators, is it peer reviewers, is it publishers? The terminology of reproducibility can vary, and that's important for us to understand as we kind of build our studies out. What is our end outcome of a study? Is it a regulatory board approval? Is it just for internal abstracts? Is it just for a quick hypothesis? That helps us both understand what sort of rigor we want in building the study, but also what kind of outcomes we want to generate to ensure we're meeting the requirements of each stakeholder's perspective. Next slide. And as we think through that, so when we're really thinking through what an epi study looks like, it's not straightforward. You have a database, you create a code and a study question, you have iterations and iterations of CSVs and tables that go back and forth on what you want, and hopefully at the end of it, you have an analytical cohort. What this ends up doing is you take a very windy road from going from data to real world evidence. So in terms of quick generations of ideas and hypotheses, this can be both a time sink and a cost sink. So what we're trying to do, if you jump to the next slide, James, is come up with a way to streamline this. So all we're trying to do with OMOP, CDM, and MEDOC is, can we come up together a streamlined way of getting from point A to point B to point C? Can we understand what questions we're asking? Can we come up with a reliable way to create that analytical cohort and then work on a way to generate that evidence for the right stakeholders? And a key part of where OMOP, CDM, and MEDOC comes in, it's going to give you the backbone of standardized data, standard analytics, and a way to present results that's scalable across different sites and make sure that we're presenting a cohesive view across all sites. Next slide. And a key part of that, and what complements and what makes OMOP the powerhouse that it is not only the CDM, but the different tools and packages that come along with it. So you don't really have to use all the tools at your disposal. The question is really what question you're asking, what milestone you want to get at. There are custom tools and packages for you to build out, but they're easily quick, standard, Atlas tools that you can really quickly jump on and present your evidence. So in addition to the scalable CDM, you also have standardized tools and packages that help you understand how to get from point A, from your data, to the evidence that you want to generate. Next slide. And this is important, and then this is something that James Brash will also talk about in the next webinars. Is as we kind of think about data, especially for oncology, it isn't one data point. There's consistent data elements along a patient's journey that need to be captured, and it's important as we build our study to understand this pathway even before we dig into the data. A way for us to capture on a person time when events happen and be able to timeline and map that out as we build analytical codes and packages is important for us to consider at the early stages as well as when we do more complex studies. Next slide. So again, going back to my first point, what do we do when we build studies? The idea is that not everything has to jump immediately to machine learning or competitive effectiveness. It's important for us to understand what we want to answer in small steps. All OMOP studies, Odyssey studies, really start off with what do we have? Like, what does the data look like? Characterizing the data, understanding the population, and then going into more estimations and prediction models. And the stepwise progress as we build the journey is important, not only for us to understand what data is needed to answer the question, but also for us to go back into our CDMs and ETL mapping conventions to really see as we build our packages and as we build our conversions, what other things do we need to consider to answer the next level of complexity. This also helps us get to what I call small wins. If you want to wait 5 years for your predictive model study and that's your end outcome is a Nature paper, that's well and good. But you're also losing 5 years of small wins that you can generate with small publications in high impact journals that can add a portfolio of things in your repertoire to really build that evidence in your journey. So that's how the Odyssey community functions, and that's how I encourage everyone to think about here as well. Next slide. So this seems very text heavy, but this builds off the previous slide. As we kind of think through going from very simple characterizations, the idea here is to how do we ask simple questions? So if you're asking for disease natural history, you really want one data element. What is your disease of interest? And as you kind of go down this table, the amount of data parameters and metrics you need also increases. So this allows us at a very early stage to think about not only the question we want to ask, but what data do we need ready across sites to answer questions. And the next series of slides kind of walk you through taking papers and kind of digging into how this works out. Next slide. So if we were to take a simple paper, so all these are publications that have been published both by Odyssey and other researchers. This study was really looking at immune-related adverse events, and they wanted to understand the frequencies of patients with non-small cell lung cancer who've taken PD-1 inhibitors and the association of that with adverse events. So if you want to click next animation, James. So what if you were to kind of create what do I need? So you really need to know what your drug is, what the PDL inhibitor is. You need to know what your adverse events are that you want to capture after taking the drug, and you need to know what the time horizon for capture is. So quite simply, rather than going and saying, I need to map every single thing in the database, now you have a very concise set of parameters that you want to ensure it's available. And the question also becomes very simple. It's amongst patients with small cell lung cancer, who are the new users of this drug, and how many patients experience immune-related adverse events within an X amount of time. So it's kind of going from an arbitrary, I have all the data in the world, to really thinking of how do I answer a very simple question. Next slide. Now, if you want to go into a bit more complex question, now this study is looking at if you were to create a prognostic model to predict 30-day mortality for CRC surgery, how do we assess with the individual social demographic, environmental factors, etc., discriminate the calibration of this model? A slightly different question, a slightly more data parameters. If you want to click next. So for this model, again, we need to know what the disease is. We need to also understand what kind of complications are coming out of these diseases. And then we need to kind of go into creating the predictive 30-day mortality model, right? So basically, your idea here is not to build the model itself. You really want to know what is the probability that 30 days after the surgery, after they've undergone for colorectal cancer, depending on these parameters available, what is your probability of getting any of these complications? Again, the idea again is the fact that you need certain parameters that came from the previous study, but now you're looking at a slightly different type of toolkit to using the same data to get a new set of analyses done. Next slide. Again, this is a higher level of complexity. Now here we're talking about characterizations of patients without atrial fibrillation prescribed any antiplatelet therapy as you go up again. So here now we're looking at longitudinal data. So far we've only had a single time point. Tell me at this point what the characteristics said. Single time point, tell me what the probability is. Now you're looking at sequences of events. Now you're looking at when someone started a treatment, where someone ended up with a treatment, and what the outcomes are. So again, at this point in time, now if you were going back to your data, you need to know if you have that patient journey that we plotted out, how complete that patient journey is. Do we get everything along that patient journey to be able to inform treatment characterizations and outcome incidents? Next slide. Again, the highest level of complexity based on our characterization, right? So now we're doing comparative effectiveness. Now we're trying to say if someone has had 2 doses of mRNA drug compared to a single dose, is there a differential risk in COVID diagnosis and COVID hospitalization? So now you need to have a bit more level of complexity. Now you need to have 2 levels of information, which is patients with and without the outcome, you need a certain more degree of parameters, because if you're doing a comparative effectiveness model, you might be thinking of a matched analysis. So if you were to go down that angle, you need certain parameters to match cases and controls. But again, to get to this point, it doesn't mean you've lost out on the previous 3 studies. It just means that as you build these 3 studies, you've added on more conversions, exceedingly more data points. But the goal is for you to build to a level of comparative effective analysis slowly over time. Next slide. Okay, so this was my takeaway slide. So the idea that I've been trying to get everyone to understand is it starts simple. Ask the simple questions, and you'll be surprised at how important simple questions can be to answer. Build complexity as you learn from those questions. Because you're part of the OMOP consortium with DIGICORE and have MEDOC, I think it's a great place for us to take that ownership, especially because the data is now standardized. It becomes really simple for you to build on the complexity. As you go back to your ETL conversions and conventions, always go back and check your data quality, because every single data point can have a different level of data quality capture. So it's important for us to not take for granted all conversions across all data points being the same. And lastly, something we all talk about is take advantage of this community. I think the key part of Odyssey, the key part of DigiCore is for us to not work alone. I think learning lessons, pitfalls other teams have could easily save time in other teams to move past that hurdle faster and smoother. I think that was the last slide that I had, James. So did I have a few more?
There was a couple more after this, just
yeah.
some of the tables, yeah.
Yes. So again, I'm not going to go through each of these tables in detail. The idea with this table, as you take your own time to go through, is when we have CDMs, be OMOP or other standard, there's obviously some limitations of using CDMs. So the way to kind of read these tables is what are these limitations when you use common data models, some things that you have to consider, and what the implications of those limitations are. And this is important as you write your paper out. So there's certain things that you want to put in your paper on what could be done and what couldn't be done. It's both important as you write that out, as well as when you design studies to understand what things cannot happen just because of the way the data has been converted to. The next slide specifically talking about OMOP itself, and I think it gives a very comprehensive picture about all the different types of data that have been converted to OMOP standards and the data parameters that are still coming up. Again, this is the same as previous slide, but specifically focusing on OMOP itself. And the last slide following that is what the community, both the Odyssey Forum, IQVIA, and DigiCore have done to kind of circumvent some of these challenges, and the tools that are available to the group and any real-world researcher on tackling some of these challenges, both currently and challenges that might come up in the future. I believe that was my last slide.
Yes, that's right. So thank you very much, Atif, for taking us through those insights. We can take a quick pause now. I do want to check if anyone's got some immediate questions that they want to ask Atif. Is there anyone? Put your hand up or shout out if you've got a question. Maybe while you're thinking of that, just one, I guess, question clarification for me. You talked about this idea of starting simple and building on from there. And in your sort of learning slide, you talked about, you build your confidence and experience, and that kind of helps you to get more sophisticated in your research. But also, presumably, there's a sense in which, as people are transitioning their kind of research data, their instance into OMOP, the fewer data elements you need to get right at the start also means you can get going quicker rather than waiting for a very sophisticated, ETL data instance to be in place.
That's absolutely correct. I think each time you bring data in, more complexity in, you have to validate it, you have to ensure it's quality checked. It has to be equivalent across all sites. So waiting for that mountaintop is great to do, but I think just like you say, James, there are small wins and things we can do in the middle that I think could easily be support and build confidence to get to the point of having everything converted.
Mm-hmm. And I think it's probably worth reflecting on here in terms of the way program structured, and what we're building towards is what these studies that I was talking about, we're really billing them as proof of concept studies. So we don't think that they'll need to be hugely complex or kind of, you know, comparative effectiveness, all of those things. We think that something that can demonstrate that we can get good, robust results around a series of simple natural history questions, I think will be more than sufficient. Now, clearly, as we move on and you guys, you know, carry on to build your, you know, research careers and build on the relationships and studies that you've started through this program, you be able to get to that additional level of sophistication. But I think that's a really good message there, and I think maybe that's something that Rosie will come to as well. So thanks for that, Atif. Were there any other questions at the moment from anyone? Okay, in which case, Rosie, tell us all about the work that you've been doing in DigiOne in OMOP.
Sure, I'd be happy to. So very briefly, for anybody who isn't aware, within DIGICORE, we have this network of centres called DigiOne, and these are a group of European centres who are working on transforming the core dataset MEDOC to OMOP. So they begun this about 15 months ago now, and we have a series of ongoing studies. I'm going to tell you about one of them today. So I work for IQVIA, but on behalf of DIGICORE as a kind of study coordinator. So we have this type of study that we call a dynasty, and that stands for a disease natural history and treatment outcome study with care quality assessment. We hope to deliver these in a few different kinds of cancers. I'm going to tell you broadly what we mean by dynasty, and then tell you one specifically in non-small cell lung cancer. So when we look at disease natural history, we are typically trying to include a very broad cohort of patients to be able to describe the means by which people are sort of referred into hospital, the way in which they're diagnosed, and any important events prior to their index date. So perhaps their prior medical history or any other cancer treatment that they received before the index date, if the index date is not the primary cancer diagnosis. We describe all of the different types of treatments that they have received following the index date, so systemic treatment, radiotherapy, and surgery. And then we also describe a series of outcomes, including overall survival, time to next treatment once you've developed a line of therapy algorithm, and in some cancers we also look at surgery outcomes, for example in gynecological cancers. And then this last objective that we call kind of care quality assessment, what we mean by that is we have taken some of the recommendations in guidelines and found a way in which we can measure them using the minimum data set. I'll come to some examples there later, but we're really looking at for what proportion of patients were the sort of guidelines followed, and if, for example, you have a certain test result, do you go on to receive the recommended treatment? And the guidelines we use are, of course, cancer specific, but we generally start with ESMO, and then we might go to some specific cancer specific guidelines such as USOMA in breast cancer. So if we go into the next slide, this one now describes the first dynasty disease natural history treatment outcome study that we're doing, and this is in patients with metastatic non-small cell lung cancer. These patients are diagnosed over a 5-year period, and we have also a lot of different subgroups of interest. So first of all, we look at patients stratified by whether or not they have metastases at the primary non-small cell lung cancer diagnosis. We also create subgroups by sites of metastases, so we're interested in looking at whether or not patients have metastatic sites in just one organ or in multiple organs, and whether or not, if it's in multiple sites, if that includes the brain or not. And then we're also looking at some outcomes by line of therapy, and then specifically we're particularly interested in those patients who receive immunotherapy as first-line treatment. So the objectives are quite similar to what I ran you through on the last slide. We have quite similar objectives across each of these studies. There's also one in breast cancer ongoing. And I guess my kind of tip to you would be exactly as James and Atif were just saying, that I mean, this has been an interesting but actually quite complicated study for us to deliver. As our first study, it was quite an ambitious number of subgroups, for example. For when you come to designing your own disease natural history study, perhaps selecting a narrower set of patients, not trying to sort of stratify by as many subgroups, and perhaps only selecting one of these outcomes. Maybe you want to particularly focus on time to next treatment or survival, rather than trying to include many different outcomes, would probably make the first implementation a bit simpler. Okay, and then this brings alive a little bit what we mean by that last objective that we call kind of care quality. So we mentioned that what we've done is we've taken the ESMO guidelines, and then we've worked through with the lead clinician and found, okay, which of these guideline recommendations can we measure with our minimum data set? So to give you an example of some things that we're not able to measure, there are some recommendations, for example, about the composition of the multidisciplinary team meeting or how regularly those meetings are held. That's unfortunately not in our minimum data set, for example, but we do have extensive information about sort of the pathology reports, about the treatments received, and about outcomes. So, for example, we can look at, if we start first looking at the recommendations related to diagnosis and subtyping of patients, we can look at what proportion of patients have immunohistochemistry for the histological subtyping. We can then see what proportion of patients have a tissue biopsy, what proportion of patients have a liquid biopsy, what proportion of patients have NGS testing, and then when we look at the testing results, we can then see in ESMO guidelines, for example, if you have a certain test result, it's recommended that you receive a certain treatment. Do those patients go on at some point to receive that treatment? So this is what we mean by measuring care quality. We can also look at the timeliness of that treatment as well. So not just were the guidelines followed, but what's the sort of distribution, for example, in initiating a targeted therapy after a test result. So these are the sort of types of the components of the guidelines that we're able to measure with our minimum data set. Okay, I'm going to go through just sort of 3 main learnings from this study, from delivering this study, which, as I said, is ongoing. So we kind of continue to learn, and we can continue to sort of check in with you as we're delivering this study. So these are things that we've sort of solved as a team, but I wish I'd been able to kind of tell myself a year ago. So the first one is related to clinical vocabularies. So when you come to forming study teams, a really good place to start is to understand for the critical variables that you're going to use in your study. Know which clinical vocabulary is used in the source data at each of the hospitals that is contributing to your study, and know whether or not that differs from the standard vocabularies in OMOP. So in OMOP, for example, when you go to search for diagnoses, the standard vocabulary is to use SNOMED, and then for cancer diagnoses, they use ICD-03. So if your source data is not in one of those clinical vocabularies, that's not a problem. There's a mapping exercise that your teams will go through to be able to map to that data model. But it is really important to know for each of the hospitals which of those kind of mapping steps has happened, because you might end up with slightly different levels of detail depending on what your source data is. So to kind of bring alive 3 examples, in the study that is ongoing that I'm telling you about in non-small cell lung cancer, we have one hospital who does all the primary recording for all their pathological data in SNOMED, and then they have all their clinical data in ICD-03. That's very straightforward. You can see compared to the standard vocabularies that is aligned. So very limited mapping has to happen there. In another hospital, they use ICD-10 for all their diagnoses, and then a local variation on histology, which they then need to map to ICD-03. But we found in the local histology kind of mapping process, we don't have some of that location information. So we were missing the topography data that is needed in some of our kind of subgroups. So we had to work through a bit of a kind of local workaround for them. And then in another hospital example, they also used ICD-10, but a different version of ICD-10 GM. So their mappings were a little bit different to SNOMED. So what had to happen was somebody's mapping the ICD-10 to SNOMED at each of these hospitals. And you'll come to know for the people in your hospitals who are doing that ETL, there is not always a one-to-one map between those kind of coding vocabularies. So there's a process you would work through to do that mapping. So in all of these hospitals, they sort of achieved it in the end, and their data in OMOP was mapped to SNOMED and ICD-03, but all by slightly different routes. So it's important to kind of know that when you're developing your code lists. The second kind of learning to share is that it again really relates to spending time to really get to know the source data at each of the hospitals that you're working with. So for key study events, such as your index date and the way in which you measure your endpoints, we need to make sure, of course, that the code lists that we develop in OMOP are encompassing all of the different ways that events are coded. So for example, if we think about the index date in this study, the index date is the date that a patient is diagnosed with metastatic non-small cell lung cancer. So about 3/4 of patients have de novo metastases. That's quite straightforward. You can use the TNM disease stage to find the metastatic date for those patients, but a bit more tricky in the sort of 1/4 of patients who have they either progress on treatment from an earlier stage, or perhaps they have relapse. So we had many different ways in OMOP that we would look for our index date in this example, and they're in the bullet points in the bottom half of this screen. So one, as I said, is we would look at TNM disease stage. That was where we could look for those patients who are metastatic at the primary presentation of non-small cell lung cancer. We also looked into the ICD-10 codes to find the metastatic codes. We had some hospitals that used them instead. We looked for the development of metastases during treatment. So as an outcome of treatment, if a patient progresses, the hospital might have coded a location of metastases, for example, a metastatic sites found in the brain or in the liver. And those location codes were another way that we could find our index date. And then in some cases as well, we can use the sort of treatment they receive to make inferences. So I guess what I'm trying to say, that while your OMOP is a common data model in the sense that you will, the same vocabularies will be used, and you'll be using the same sort of tables in OMOP, you still need to be conscious of the fact that, as we all know, cancer data is very complicated, and there are many different ways that you can code an event. So you need to make sure that you've incorporated all those different ways in which it can be coded into your definitions. And the last one is really sort of a data quality step. So we're all very familiar with sort of applying inclusion-exclusion criteria from protocols, and we need to find a way to translate our protocol into a script so that we can find the eligible patients. But one thing I would really encourage you to do for good practice as a data quality step, but also it's important when you come to publish, and many journals want to see this, is understand what the attrition is at each criteria that you apply. So for example, in our non-small cell lung cancer study, we begun with looking at all lung cancer patients from November 2018 onwards. We then had to make sure that we excluded anybody who had said they didn't want their data used for research purposes, not able to analyze those patients' data. We then needed to find only the non-small cell patients, so exclude those other forms of lung cancer or any patients who have mixed histologies. We then needed patients who had a tissue diagnosis, etc., etc. You can continue down this slide. So these are the inclusion-exclusion criteria, and we went through a process with our contributing hospitals. In fact, we're still working on it currently. It's in progress of seeing what the attrition is. So the proportion of patients that drop to each of these criteria. It's a really good exercise to do, as we actually realized that we had some quite different drops at different criteria, and you would really want to question, unless the hospitals that you're working with have a really different sort of treatment pathway, probably quite unlikely. You would not expect to see massive differences in the number of eligible patients that you have compared to the sort of top of the funnel, the top of your inclusion-exclusion criteria. So as an example here, imagine if across 3 hospitals, your criteria eligible find 6% of all lung cancer patients in one hospital and almost a quarter in another. I mean, that would really need to be sort of investigated. So this is a really kind of good practice to go through. And as I said, also for when you come to publishing, many journals will want to see the attrition by each of your protocol criteria.
Brilliant. Thank you so much, Rosie. That was really insightful and helpful. Again, an opportunity for anyone who's got any questions. Have a little think now if there's anything you want to ask. And Rosie, I'll have a question for you while people are maybe mastering their thoughts. So just cycling back to Atith's presentation, there's a key message there around, starting simple. And I think that came through really nicely from you, that, with that natural dynasty study, there was perhaps, a simpler starting point you might have started from if you'd wanted. The other thing I was reflecting on is, and this was a message that we heard today and we heard last week, about this idea that there's this community out there, there's Odyssey, there's all these tools, there's code and everything else. I wonder if you could reflect on where you've either benefited from that and been able to bring in, stuff off the shelf that people have developed before. Oh, and maybe the flip side of that, the bits that you've kind of, for this study, you've really had to invent and sort of, or kind of build from scratch. To what extent has that community given you a head start here?
Yeah, absolutely. There's been we've used a few, certainly when it comes to the sort of characterizing your patients around the index date, the sort of baseline characterization, and looking at kind of treatment patterns. We've been able to use some of the Odyssey tools there. I guess when we've had to create something a little bit more bespoke was, for example, if we're looking at line of therapy algorithms, as you can imagine, are very cancer specific. You're probably scripting out something very specific there, or are looking into our kind of guideline compliance. That was a bit more unusual, but certainly there are sort of cohort diagnostics types packages and treatment packages where you can really start as a group, as a first step to really kind of explore your data. That's really helpful early on when you're doing some of that comparison. Are we seeing a similar kind of cohort? Are we seeing similar size of subgroups and things? It's a really useful first step. And then maybe to address perhaps all of your protocol criteria, if you've got quite a specific research question, you might have to script that out a bit separately, but it's a really good starting point, as you say.
It's nice to hear how that really has been a reality for you, that kind of head start. Okay, thank you. Any questions from anyone else? Aristotle.
Hello. Well, I would like to ask what happens if your data are in ICD-10 and not in ICD-03? Who is going to transform the data from ICD-10 to ICD-03?
I mean, this is...
So this, well, I guess, obviously, let's, bearing in mind that this program, this training program, is very much tied to the I3, the I3 project, the DIGICORE I3 project. So for sites that are in that program, there is resource already in place, as I understand it, to support that extract, transform, and load. And we also have a parallel training course, which is starting to help people with that program. I mean, in terms of the specific, I don't know, Rosie, if there's anything specific to add, or indeed James Brash, in terms around what we've been doing in metastatic non-small cell, if we've had that particular challenge.
No, you're absolutely right. I mean, for each of the hospitals in DigiCore, there was somebody who was kind of dedicated to doing that mapping exercise, as you say, James. So to going from source data to the kind of vocabulary in OMOP, and I mean, crucially, the kind of clinical inputs from each of the kind of clinical leads to help them through that was needed as well. So yeah, agree with you.
Okay.
Is a medical doctor going to be needed for this, or?
We've really benefited from that expertise, haven't we? Not throughout, not at every stage, but there's been key points where we've absolutely needed that input. Rosie, you know, when, for example, when we were together working on this study, helping us with both the interpretation, but actually also the correct allocation of particular patients to particular subgroups so that we could accurately do that.
That's yeah, absolutely right.
Thank you.
James B.
I just want to add on to that comment. So in regards to the support from clinicians in this process, they have been fundamental in us and putting together these studies and running these studies, and I don't think we'd have been able to do it without their input. But the actual OMOPing process, which I think you started with the reference there to Aristotle, so whether you either go from ICD-03 and map that, or you start from ICD-10, or whatever coding system you're in, at this stage, there's enough conventions and documentation in the community that you can map those over without a clinician oversight now.
That's really helpful, James. Thank you. I think there was a comment in the chat as well, OPBG, essentially what I was saying earlier about, you know, getting to know each other and relying on each other. Then there may be folks on this call who can help with that kind of process. Any other questions? Atif.
This isn't a question. This is more of just a general comment. I think the point that Aristotle raised is very important, and is going back to what I was trying to talk through my conversation as well. Is when you kind of craft your question, I think it's important to understand: do we need that detailed histological, morphological map up when we want to answer a question, or are we just looking at broad characterizations at a very diagnostic scale? So I think it's important. Again, obviously, it doesn't mean that, just like James said, we're not converting, but the scale of conversions, what detail do we need, where we're getting at, also points us towards how much level and effort do we need to put in to get to answer the question at hand. I think that also allows us to say, as we plan forward, if you want more of these detailed questions, we can prioritize effort in some of these high level conversions first before digging into everything else. So it is a cycle. I think it's a back and forth conversation, just like we're all saying. How we do study informs how our conversions work. How our conversions work also inform what kinds of studies that we do. So this team effort of ETL, data scientists, researchers, clinicians work very closely on moving the ball forward for us to start answering more complex questions. But it doesn't mean every single question needs complex data.
And I think that's maybe just before we move on to the wrap up, just a point that's worth reflecting on, that multidisciplinary sense of clinicians, data scientists, biostatisticians, and everyone. And of course, the people that we have on this call today and on this course represent that diversity. And that's very much a deliberate kind of choice of how we've done this program. We're wanting to bring those different skill sets together. So actually we can have the right skills in the right teams to drive this research. So thank you very much. I'm just going to suggest, as we move towards the end, it would be worth taking a couple of minutes just to introduce you to the SharePoint. And Will, are you able, is that
James,
actually going to work?
Yeah, there's one more question, I think, from Tamsin in the chat.
Oh, sorry, I missed that. Tamsin, hi, I beg your pardon.
Hi, no, that's okay. Thank you so much. I just wanted to ask a question to Rosie about this concept of defining measures of treatment quality using kind of routinely collected data and data that's available within your sort of minimal data set, because I think that's really, really interesting and important. And I'm curious about, in your study, to what extent is it sort of acceptable to use those measures on the assumption that it's something that we measure and it fits in with guidelines, and therefore if it fits in with guidelines, then that means that it was a good quality treatment, and if it doesn't, then it wasn't? Or is there kind of a further sort of process in the analysis of linking that to the patient's actual clinical outcome that needs to happen during that study to show that yes, that patient's treatment was effective, or it wasn't effective because that sort of parameter was or wasn't met, if you know what I mean? So I'm just kind of curious about that process and how that worked, what is working in the study.
Yeah, that's a very, very good question. So actually, you've just raised what we really want to do as the sort of next step. So you're absolutely right that, well, first of all, that the fact that the patient didn't receive the ESMO recommended care, of course, doesn't mean that the care was of poor quality, because there may be many reasons that the hospital was not able. There are, of course, many hospital level structural barriers to hospitals being able to deliver it. So we're going to need to be very careful in the interpretation that, you know, if there's a hospital, for example, that doesn't have access to a treatment, of course, that's why they're not prescribing it, not because they just, you know, made a personal choice not to. The really interesting follow-on question that we hope to do in a kind of next study is to then, exactly as you say, is to link some of these metrics to patient outcomes. So actually in the first study, we're only looking at did you receive the ESMO recommended care, and to characterize the patients who did and didn't, try and get some sense for perhaps why perhaps they didn't. And as I said, looking at time to care. And then the really interesting question after that will be to look at, for example, does time to initiating treatment have an impact on survival, say, which we would imagine it would, to be able to link that to patient outcomes. So that's not part of the first study, but is a really interesting follow-on kind of question to look at, exactly as you say.
But it's interesting to know that you know, just asking that one simple question is, like you were saying, just asking the one simple question using the data that you've got is enough to have kind of a robust sort of framework for doing your analysis, and then the more complicated question comes after, maybe.
Build on that, exactly, yeah.
I'm just interested by and I found myself saying things, using the word simple. And actually, it's often, even the things that we sort of blithely say are simple are actually really interesting, quite complex, and they raise quite tricky questions. So, yeah, even things that look fairly straightforward can be absolutely fascinating and difficult.
Thanks.
Yeah, just like James said, I wouldn't equate simple to not important. All questions are important.
We don't see your screen any longer, Will.
No, I'm going to share again. That's fine.
Okay.
So very quick wrap up. Thank you, our presenters today. We're aware through this, James said it at the beginning, that there's a lot of information here. And as we get towards building studies, what we plan to do is to help package this so that you have a very clear guide around which to build your studies, with a reminder of this material that's being presented here. But I just want to share the SharePoint. Hopefully you can see that on the screen. Mm-hmm. You will be receiving an invitation to this. Please do check junk mail if you haven't received an invitation in the next couple of days. I'm hoping it'll come tomorrow. It may be on Monday. Please do get in touch with the training email address to say. There's often some to and fro on some permissions, sometimes with some domain names. But this is the IDEAL4OMOP SharePoint site. So I'm just going to briefly show you this. There's an introduction to the course, and here you'll have news items which should come through on your email every so often. We'll just remind you of progress as we go through the program. The most important probably will be the course materials, and when you access the course materials, you can see here from the first webinar there'll be a recording of the session, and there'll also be the slides in PDF, and then the further reading that might be mentioned in the session, web links and other things to to go away and read. And we do recommend you trying to clear some time between sessions. That there's only so much you can take in an hour of a webinar, but really it's digging into the material between times. Having said that, we do have a kind of quite a brutal time scale in these 2 weeks. We have our next one next week. As we go through, those sessions will be spaced out a little bit more and there'll be more time between. But as I said, please don't worry about capturing everything in these hour calls, because the SharePoint will be there and you can come back and refer to the material in whatever way most suits. So the best way, if you go back then to this IDEAL4OMOP menu, it'll take you back to the home page. We've got a frequently asked questions list there as well. You may have questions about DIGICORE and the program. Please do add to that if your questions aren't recorded there. And then the course overview really is just that introductory webinar. If you missed it, there's a link there to the DIGICORE YouTube and a brief overview of the course. Crucially, the SharePoint will begin to be the place you build your collaboration. So this will be a place where you can start to share draft protocols and ideas between each other. We're hoping to bring some Teams, Microsoft Teams community through this as well. So it'll also be a way to communicate with each other. All that to come. It'll continue to develop. The first step is to get you all enrolled in it. And as I said, all the course materials will be there for your reference. They'll also be listed in the documents there, and a little reminder of who the Leadership Advisory Board is, and a lovely cheery photo of James Anderson, your course instructor. So keep an eye on that. Do come and have an explore, and as I said, keep an eye out for the invitation email, which will come in the next couple of days. And please do get back with anything else that you'd find helpful to go on there. Thank you very much.
Thank you. We're just up to the hour, and I will just end by reiterating two things that Will said. Number one, thank you very much to Rosie and Ateep for some fantastic, really thought-provoking insights onto what's possible and also the challenges of working in OMOP. And secondly, what Will said around, do reach out to us if there's things you want to know, things you're not clear about. We can tailor this course as we go along and look to bring in some additional expertise, but hopefully things are starting to materialize, things are starting to come together. That's all from us. Have a great day, have a great weekend when it comes, and we'll very much look forward to seeing you pretty much this time next week. It's an hour earlier where we'll have the third seminar. So thanks very much.
Thanks, James.
Thanks, everyone.